introduce some basic biological concepts in root development. We talked about specific locations, the steps that are involved after the wounding process occurs. Then we went on and discussed some other concepts that are related to plant biology that would impact plant propagation, from polarity, the top side and bottom side of the cutting, the proximal and distal ends. We went on and talked about correlative effects in how one plant organ has control over the development of another plant organ. And so these are some of the specifics of some of the steps in root formation in cuttings. And we'll advance on with that now. So as the plant propagator is taking cuttings with this goal of maximizing rooting in the shortest period of time, our goal is to end up with this, a rooted cutting. And of course the soil's been washed away so we can see the vigor of the roots, the proper color. They're turgid, and that's what we want. This was placed into the proper environment, both in terms of the soil and the air surrounding it, and there was rapid root generation. And so then at this step, the propagation manager would send it off to another department, so to speak, the production department, and the production manager would transplant it into a larger pot, or maybe it's going to be packaged and sold to another nursery, similar to what we saw in Magnolia Gardens Tissue Culture Lab, where most of their rooted liners were boxed up and sold. So we have before us a rooted liner, L-I-N-E-R, rooted liner, and it is ready to go to the next production step. So this leads us to a question: where did the cutting come from? By and far in today's nursery industry, most cuttings are secured in-house from your own stock plants. Your own mama plants, your own stock plants, plants that you're typically growing and growing to a stage where they will be sold. At the same time, during this stage of growing up to the finished point, there's coordination between the production manager and the propagation manager. And these plants that are being grown for sale are also a source of cuttings. So long gone are the days where we had specific plants just planted in the ground, and that's all they did was just serve as a source of cutting material. Not too much of that remains nowadays. Again, by and far, it's your own production of plants that you utilize as a source of propagules for propagation. Again, that takes some good coordination between the production manager and the propagation manager. So considering the wholesale plant industry, great coordination here. The production manager essentially gets permission from the production manager to send workers out into the field, into specific blocks of specific varieties of plants, and takes cuttings of them. And those cuttings, that's, a couple of things are happening there. One, it's helping shape the plant as it continues to grow and mature into a final saleable plant. But those trimmings, those stem cuttings, are a source of propagules and has value to that propagator. And of course, they are typically collected into buckets and hauled back to the propagation facility, the propagation greenhouses. So the plants before you that are being grown for sales, they also serve as the mother plants or the stock plants. And again, there's concern here with the propagation manager for the health of these stock plants. They need to be well watered, well fertilized, good nutritional status, free of insects, free of diseases. I think many times, especially in the warmer months of the year, propagation managers try to send their cutting crews out into the fields to secure cuttings early in the mornings when there's less heat stress and in turn water stress on the plants. The cuttings tend to be more turgid, greater water potentials in that tissue, and just overall better health in the cuttings, and there's less stress on those cuttings if they're harvested in the morning. So they're collected up out of the field, labeled, and sent back to the propagation facility. Typically some greenhouses where they have intermittent mist set up and control over the light and temperatures. So moving on here, are there ways we could consider to manipulate the stock plants to our advantage to maximize cuttings? And why would we even consider that? There are some plants that root easily. You take cuttings, you stick it right into the soil, maybe even without rooting hormone, without additional auxins, and they root easily. But most of our plants propagated asexually do not fall into that categorization. They are a bit more difficult to root or difficult to root. And so then that leads us to this question, is it possible to manipulate the stock plant in such a way to yield more cuttings and cuttings that root easier? So let's try to summarize some of the information on how we can manage stock plants to either maximize the number of cuttings or maximize the speed at which those cuttings will root in propagation. I think this begins with a discussion on, maybe just select easier to root species. If I'm growing a crop that doesn't root easily and it remains in propagation for months at a time and it roots slowly into the container, that's cost that's added to that product. Is it a better decision just to not grow it? Let somebody else grow it. So that might be a good place to begin, in industry at least, is to just make a decision whether or not to grow it based on its ease of rooting. So then beyond that, we might have plants that root moderately well to still difficult. Keeping those plants, those mother plants, in a state of juvenile growth or juvenile transitional stages increases rooting potential. We want to maintain those mother plants, those stock plants, in a state of juvenile growth. If you think back to the life cycle of the plant, whereby we describe a germinating seed that enters now into the juvenile phase, eventually a transitional phase, and eventually that adult phase where it's sexually mature, and then flowering once again, continuing a new life cycle. Those phases of the life cycle, coming back to that juvenile stage, that tissue tends to be easier to root, and in some instances much easier to root than the adult tissue. So this isn't necessarily chronological age. The new growth that's emerging from the shrubs right now over the last 2 weeks, that isn't necessarily juvenile tissue. Chronologically, it would be new tissue. It's just emerging this week and last week. Here's another explanation of this comparison of ages in the tissue of plants. Okay, think about your hands. You wash them several times a day to get rid of germs and viruses. Lots of soap and water on there, and skin cells slough off. And from those basal skin cells, they're constantly dividing, giving rise to new skin cells that move up towards the surface. And as they age, they slough away as you brush up against things or as you wash your hands. So regardless of your age, me, you, other people, our skin cells on the surface are 3, 4, 5 days in age, right? Because they're being sloughed off so rapidly. Our skin cells on the surface are 3, 4, 5 days in age. That is a chronological age. My skin cells, your skin cells are 3, 4, 5 days old. That is a chronological age. However, that is different from physiological age. As you examine the two hands in this photograph, one's from a very young person and the other's from a very old person, you see differences in the skin cells, don't you? So even though both of their hands, their skin cells are 3, 4, 5 days old, chronologically, physiologically, there's a big difference. The hand on the right, those skin cells are physiologically mature. The hand on the left, those skin cells are physiologically juvenile. So I'm making this analogy here for you to think about the location on the plant and its growth phase, not necessarily chronological age. Not that growth that's emerging from plants right now. It may or may not be juvenile growth. But the location on the plant might be different in terms of its physiological age, juvenile versus adult. So again, coming back now to stock plants out there in the nursery, if we can consistently cut on them, and that cutting and trimming leads to more branching, that cyclic action of cutting on them tends to keep them in a juvenile state of growth. And that's good because, again, that juvenile wood roots easier than the mature wood. So this is part of the key to maximizing the production of cuttings and the speed and ease of rooting in those cuttings. So again, the difference in chronological age versus physiological age. Okay, what else can we consider in maximizing cuttings in our stock plants? Okay, so you just saw the first two bullets there. We also have some evidence that we can manipulate stock plants by cytokinin. Remember the hormone cytokinin, we can dilute it in water and apply it to freshly trimmed plants, where the buds are removed from trimming them, and you spray on cytokinin. The new growth as it emerges exhibits greater juvenility. So yes, this has been demonstrated in some plants. Greater juvenility once it's trimmed and afterwards treated with a cytokinin solution. Something similar happens with gibberellic acid applications. Plants that exhibit mature growth, they're trimmed back and immediately sprayed with gibberellic acid. And that sequential growth that emerges after that GA spray, that growth exhibits juvenility. Another way to rejuvenate or induce juvenility in stock plants. Many other plants, it's the method of micropropagation. In the methods of micropropagation, those plants tend to be in the juvenile state. And as they grow up and mature and go out into the field, they continue to be in that juvenile state and exhibit good rooting potential. So yes, we have some of that knowledge at our disposal on how to rejuvenate existing stock plants that are physiologically mature. And we also have some creative ideas. It's some information that comes from etiolating plants. Etiolation is the development of plants in the absence of light. So maybe think of it as the plant is growing and all of a sudden you remove it from the light. You place it into darkness or into extreme shade. And that absence of light causes some physiological changes. And in turn, this is demonstrated as a way of inducing juvenility, greater rooting potential in cuttings. Okay, so etiolation in a plant, if the entire seedling, as an example, is etiolated, it's tall, it's spindly, maybe there's too much competition from plants around it, it's not getting enough light. We sort of saw this in our light experiment with those seedlings that were in the low light. They were weak, they were tall and lanky, skinny, as opposed to those grown in a higher concentration of light. So I'm trying to paint the picture for you in your mind's eye of an etiolated plant. Or again, maybe the plant has matured and all of a sudden we place it into darkness. And once in that darkness, the plant continues its attempt to grow and survive, and that subsequent growth tends to be easier to root. Along those same lines, there's a technique known as banding, and I've got a couple of photographs to show you, but it's just a localized exclusion of light. Okay, coming back to physiological age and that location on the plant. So this might be demonstrated like this. We have a plant that's, maybe it's a tree, and it's growing out in the field, and we cut it back hard and like almost to the ground. And it emerges from that stump and grows for a few years, maybe 3 years. And as that tree grows up taller and taller and taller, that newer growth at the top of the plant, so if you look towards the left of the screen, that tallest tree that's in the wintertime and it's leafless, the tallest part of that tree, yes, chronologically, it is the youngest. It's probably 1 year old. However, physiologically, it is mature, and its rooting potential is low. Whereas at the base of that plant on the left, the physiologically juvenile wood is at the base of that plant. So it's not changing, it's not moving up the plant, it remains at that same location, in other words. Okay, so it's, again, you may look at, and this actually frustrates a lot of home gardeners because they have this maybe beautiful shrub in their yard and they, oh, I want to take some cuttings of it and make some for my friends and neighbors. And they go and take cuttings and nothing happens. Or they have very poor rooting and they don't know why. They thought they had a green thumb. Well, they're taking cuttings from their favorite shrub and it tends to be mature wood. Okay. So as you look around you right now, a lot of our trees and shrubs are budding out. That's new growth that's emerging. Very young wood, yes. Chronologically, it's only 2 weeks old. But physiologically, it may be very old. Okay. So again, we think about this also in terms of where we collect our cuttings from on the plant, and how that's influenced by the physiological age. We talked about etiolation. So here, this is at a nursery in England, and they have some apple rootstocks that they take cuttings from the trees, root them in the ground to serve as the rootstock for grafted apple trees. And they build this structure right over the stock plants out in the field. It's made out of 2x4s, and then they place this black plastic over it, staple it down, tie it down, and this structure excludes all light, and they'll keep it on there for, in this instance, about 5 or 6 weeks. So the apple trees that are inside there, total light exclusion, they're trying to survive. They're going to extend their shoots out, trying to find light. And the research demonstrates that after I take this structure off here and I go in and harvest that growth that has emerged in the previous weeks, it demonstrates better rooting potential. Great, yeah, I can take my cuttings and they root faster, more vigorously in the greenhouse. More precisely, there's some evidence that shows that in that etiolated tissue, that stretching material, that there's a buildup of starch grains, and that may be part of the relationship to more rapid and successful rooting. So etiolation, the exclusion of light from the plant. And along those same lines, we mentioned a special type of etiolation known as banding. And this may be a strip of black tape or black plastic or Velcro that again is located on the stem for a period of time. You would place this material on the stem where you plan to cut it. Okay, so on the right there's a redbud tree and there's a piece of Velcro tape on it. That will remain there for 5 or 6 weeks, and then that Velcro will come off, and I'll cut it away from the mother plant just right there at the bottom of that Velcro tape. So all that tissue that's in darkness right now will exhibit a greater potential to root. So a banded stem or localized etiolation. So these are some of the methods we have at our disposal, some techniques we might utilize to increase the rooting potential of those cuttings taken from our stock plants. Some other methods that have been demonstrated to induce rejuvenation, and maybe it's serial grafting. And so the story goes like this. A tree is identified as an elite tree. It just has ideal branching characteristics, maybe other phenotypic characteristics that are deemed superior or elite. The problem is now the tree's age. It might be 10, 20 years old before you discover it. If you take cuttings out of the canopy, physiologically it's a mature wood. It's not going to root or root poorly. So we're going to attempt to induce juvenility. So the way this works is we'll start with a rootstock. And by the way, this is eucalyptus. It's a native to the Australian continent. Beautiful tree. And so here's an elite tree, and we want more of them. So we take cuttings out of the canopy and we graft them onto another eucalyptus, encourage it to grow. It branches. We may pinch it, trim it to encourage rapid growth and branching. And so then we remove a cutting from that canopy and graft it onto another plant, grow it out. And again, it's grafted at this point. And we take a cutting now from, and I think it's important to remember here, the genetics, since it's a grafted plant, the genetics are the same as that elite tree that we started with, even though it's growing in this bush form now. Okay, so we take a cutting, we try to root it, it fails. Again, we take a cutting out of that canopy and graft it onto another eucalyptus, grow it out into another shrub. This is round 2. Take a cutting, rooting fails. We repeat that process 6 times. And with this particular eucalyptus, it wasn't until round 6, the 6th-step graft, that finally enough juvenility had been induced in that plant that it successfully rooted and grew on its own from a cutting. And again, it can be trained into a tree form, but what's important here is now we have a shrub form of this plant. Genetically, it's the same as that elite tree, and we can take numerous cuttings off of it, root it, train it as a tree, and that was our goal. We achieved our goal through this serial grafting. All right, moving on here. Some other techniques at our disposal to increase rooting potential. One might be wounding. So yes, the cutting of the stem away from the mother plant is a wound within itself, but increasing the wounded surface is an additional technique. And so here the propagator will take a knife and scrape off part of the epidermis all the way down to the cambial tissue, increasing the size of that wound before it's then dipped into a rooting hormone and stuck into the soil. Again, we have evidence that increased wounding can increase the rate of endogenous hormones in photosynthates to that wound location and speed the rate of root formation. So yeah, additional wounding. Again, for the propagator, you have to weigh this out. That's an additional step. It requires more time per cutting. but of course, if it leads to improved success in rooting, it may be worth the time to take this extra step. All right, in this instance, those are Taxus cuttings. It's an evergreen shrub, sometimes tree, and this works well in junipers also. As a matter of fact, you all did some of this in your lab, and those cuttings are still in the Interstate 45 greenhouse. All right, moving along here, some other techniques that might improve the rooting from cuttings coming off stock plants, and one is girdling the stems. So here the idea is we have our stock plant and we examine it for potential cuttings in the future. And what I'm going to do is identify those stems and go down from the tip maybe 6 or 8 inches, generally 6 to 8 inches down from the tip, and girdle the stem with like a bread twist tie or maybe a piece of plastic, and it's not so deep into the tissue that it restricts the water flow through the xylem system, but it is enough of a constriction that it restricts the flow through the phloem system. Okay, so think back to your plant physiology. The phloem tissue translocates photosynthates, generally from mature leaves down to the roots and other metabolites. And by girdling that stem, again, we haven't cut off the water supply to the tip of the stem, but just enough to girdle it and prevent the flow of that phloem downward. So maybe in your mind's eye, think of a stem, and 6 inches down, you put a twist tie from a bread wrap or twist tie on it, and you come back 2 to 6 weeks later. Well, just above that bread wrap or twist tie, it's going to be swollen because you've prevented the flow of carbohydrates and other metabolites downward through the phloem system. And so then I would then cut it away from the mother plant, dip it into rooting hormone, and that girdled stem, those tend to exhibit a higher rooting potential. Again, another method of speeding up the rooting process. Because again, that's my goal as a propagator, is to grow roots as quickly as possible, get them out the door, and start sticking other cuttings. Going to this next bullet here, that of carbohydrate supply. So carbohydrates are not directly linked to rooting. Remember, we in our last lecture talked about the steps that lead to root formation, and it's heavily relying on auxins. Okay, so carbohydrates, are they directly linked to root formation? No. Is there a correlation between carbohydrates and root formation? There is. So maybe think of it like this. That cutting, if it has a good supply of carbohydrates, it's most likely going to root easily, because those carbohydrates are building blocks for growing new cells in those new root organs. Okay, it's not directly related, the carbohydrates are not directly related to the root formation, but there is a correlation between carbohydrate concentration in that stem and the success and the speed of root formation. So again, it just comes back to having healthy stock plants that, growing out there in the field, perhaps they've been well watered, well fertilized. They don't have any type of environmental stress placed on them. And you would expect those cuttings then to have adequate supply of carbohydrates. So moving on now from carbohydrate supplies. So we've given you some ideas here on how to manipulate stock plants to your advantage? Are there specific locations on the plant, or maybe time of year, that need to be considered also, so that it influences my success? So let's try to summarize the selection of cuttings on the stems of those stock plants in such a way that I can kind of put heads and tails to it and improve my success in rooting. So we're going to think of it this way. In woody perennial plants, woody shrubs, trees, we can summarize it like this. Let's first compare differences in lateral shoots versus terminal shoots. The rooting differences between lateral shoots and terminal shoots. Okay, so just a quick refresher. Think of a woody shoot. The terminal bud, that is the terminal shoot. It's out there at the tip, the terminal. Up and down that branch, there are lateral buds that will come out of dormancy and give rise to lateral shoots. So are there differences in rooting potential as those two types of buds resume growth in the springtime? So generally speaking, it goes somewhat like this. If I'm taking a softwood cutting in the springtime, generally the terminal shoots root better. If I'm taking a semi-hardwood cutting later in the spring or the summer, generally it's the lateral shoots that exhibit greater rooting. Okay, and so if there's a specific plant and you're not sure, maybe you've never propagated it before, maybe consider that in terms of, the season of the year, the location on the stem. Is the stem I want to cut from a terminal bud, or is it from a lateral bud? It might make some differences in your rooting success. What else as we consider woody perennials? Perhaps differences in various parts of the shoot. So think of a shoot that's, 1.5 feet, 2 foot, maybe 3 foot long. If I cut it up into sections, which sections might root easiest? Would it be those at the base of the shoot, or those way out at the tip, or perhaps those in the middle? So again, generally speaking, as you consider those various portions of a single shoot that might be 1 or 2 feet in length, the basal portions, you've got this long shoot that's 24 inches. If you come back to the base of it and start cutting those into pieces, those tend to root easier than those pieces you would harvest from the tip in most plants, okay? And again, that's a generalization. So maybe think about that also as you start harvesting wood out in the field and cutting them down into pieces to propagate. What's the location on the individual shoot? Is it way out towards the tip, or is it towards the base, or perhaps the middle? That might influence my rooting success. What else might we consider? Is the wood in a stage of flowering, or is it still vegetative growth? So vegetative wood, we mean it's growing leaves and shoots and buds, whereas flowering wood, well, that's fairly specific and discernible. What typically happens if you attempt to propagate flowering wood is it just tries to continue to flower, and there's very poor rooting, and if it does root, that newly rooted plant tries to continue to flower. There was one semester, I taught this class in a summer session. We did this with crepe myrtles, and we took some stems of crepe myrtles, and that's exactly what happened with the shoots that were harvested right near the flower. They rooted generally well, but they just continued to try to flower. Whereas those stems further, those pieces further down the stem towards the base rooted much easier. Okay, so generally speaking, we try to avoid flowering wood. Maybe you also consider the seasonality. Is the cutting I'm taking from this specific plant, is it in the winter? Perhaps that new growth in the spring that we're seeing right now that's soft? Or maybe it's later in the summertime. The seasonal timing also imparts differences in success with some of our species. For example, there's some shade trees that, believe it or not, you can take cuttings from them when they're in dormancy in the wintertime, take them into a greenhouse, and they root rather well, as opposed to that same species, if you take cuttings in the springtime from that new growth, it has a very low rooting potential. And so with each species of plant, if you're not certain and you can't find an answer from somebody, you would just have to experiment by harvesting cuttings in various seasons of the year to make some determination on how seasonality impacts the speed and success of rooting in that species. So some more considerations here in our stock plants that influences our success in propagation. There are instances where you have cutting material, either you've purchased it in and you don't have time that day, maybe not even tomorrow. What do you do with all those cuttings? Or maybe the production manager tells you, we're going to be cutting on these red tip photinias, you want the cuttings? And you say, you bet I want them, but I can't stick them today, my workers are busy. So you might be faced with a situation where you have to store those cuttings for a period of time. And when I worked in industry, I did this all the time with some woody cuttings and some herbaceous cuttings, sometimes for up to 7 days. And, maybe think of it this way. If it's a plant that's from the temperate regions of the globe, in other words, if it's from a region that freezes in the wintertime, you're probably going to want to take those cuttings and store them in refrigeration in the upper 30s, maybe low 40s, keep them moist. And you might be able to store them for, if you do this in the wintertime, for a couple of months. If they already have leaves on them, it's probably going to be a shorter time, but nonetheless, the key is cold, moist conditions. You're slowing down respiration, reducing desiccation as you keep them in cold, moist conditions. Now, if it's a plant that has its origins more in the tropics, where maybe it doesn't freeze in the wintertime, likewise, you can store those for a short period of time. With those plants being stored that have tropical origins, maybe think about storing them in refrigeration that's more in the 50-degree range, 50 to 60 degrees, and likewise still moist conditions. Again, you're reducing respiration, trying to reduce desiccation. And in those storage units, of course, you want to make sure you stay on top of any type of diseases that develop, because that can quickly spread through those cuttings that you're storing. And if it remains in there, you're propagating the pathogen also. So yes, that is possible to store cuttings for a period of time. Some other considerations here in rooting, and that is mineral nutrition. So this would be nitrogen, phosphorus, potassium, calcium, sulfur, on down the list of all 17 required elements for that plant to complete its life cycle. And yes, some of those minerals are needed in higher concentrations like nitrogen and potassium. So as we start sticking cuttings and encouraging roots, do we immediately apply fertilizers? Do we immediately introduce nutrients? And it's always been a topic of debate, and it's still being researched, but generally speaking, no. If I harvest cuttings from healthy stock plants, there should be enough carbohydrate supply and mineral concentration in that cutting to satisfy the demand of new root initials, those new root organs that are forming. And once that cutting is rooted out into the surrounding soil, then I can consider or that's when I should consider the addition of mineral nutrients. Remember also in propagation, it's very easy to leach and lose nutrients, and that's just a loss of money. You could potentially harm the environment, especially with nitrogen and phosphorus. So this is somewhat tricky. You really have to think about the plant health, the stage in which it's rooting, and then start introducing the nutrients in the form of a fertilizer at the appropriate time when you're certain that nutrient absorption is going to occur. All right, folks. So think about all those aspects of plant manipulation and improving rooting. If you have questions on any of those cutting types, locations, manipulation steps, please let me know. Y'all have a good day.